Hello, students. A very good afternoon. So let us talk about the final part of our human health and disease. So that is we have to talk about HIV or AIDS, cancer, and finally drug and alcohol abuse, so that we can conclude this chapter finally. All right. So let us start our topic today. So yesterday we were discussing about AIDS, right? So AIDS is acquired immunodeficiency syndrome. I had mentioned. So, anyways, before we start the topic in detail, let me launch an NPS poll. Please let me know if the audio and video is clear. All you have to do is let me know if the audio and video is clear. YouTube students, you can just put a message in the chat box. All right, so let's end the NPS poll. I hope everything is clear. If there is any problems, please let me know. We can just sort the problems as soon as possible. Okay, fine. So 1 second. Here we go. AIDS or HIV. Okay, so we know that AIDS is a disease. Okay, and the full form or abbreviation of AIDS stands for acquired immunodeficiency syndrome. So it is an immunodeficiency disease. Okay, so whenever you lack in immunity, there is possibility of you getting infected or you being vulnerable for many of the common diseases that we have the capacity to we have the capacity to protect ourselves from. For example, it might be tuberculosis, cholera, diphtheria, okay, and common bacterial and fungal infections. So we have the capacity to protect ourselves because our immune system is strong enough to do that. Whenever if there is an impairment or if there is any alteration to immune system such that our immune system cannot function and complete its mechanism, at that point our body becomes defenseless. When our body becomes defenseless, then what will happen? Our body is prone to multiple diseases. So you just imagine yourself, whenever you were whenever you were infected by a certain disease or a bacterial infection or a fungal infection or a viral infection, did you get two diseases at a time, like dengue and typhoid together, or malaria and dengue together? Okay, it was not happening, right? So similarly, when we have in a normal condition, it's impossible to get more than one symptom of a disease. Okay, apart from certain viral infections such as AIDS and COVID-19 or something like that. So that is why our AIDS shows multi-symptomatic condition where a person will start experiencing or showcasing more than one symptom of a particular disease. Okay, so that is why we call it as a syndrome. All right, so AIDS is affected. I mean, like, we get AIDS because of a virus called as HIV virus. HIV stands for human immunodeficiency virus. Okay, so this HIV virus is a retrovirus with an RNA genome, and it was first reported in USA in 1981. So till then, in 25 years, it's very astonishing to just to look at the numbers, because it has claimed around 25 million people's lives. Okay, so it is very high number for a disease. You might tell me malaria claims even more bigger number. Why? What is so special about AIDS? Why are we giving more priority about AIDS? So if you see the way the HIV spreads, okay, it is quite difficult to even imagine how is it even possible to achieve such numbers of infection when it can spread only through a fixed ways of transmission. Okay, so let us see how is it possible for a person to contact HIV. Okay, So HIV, a person can become HIV infected where through sexual contact with infected person. So it's clearly mentioned only when a person, okay, have a sexual contact with an infected person, the other person, the normal person can become, HIV positive. All right. So next one is transfusion of contaminated blood. So usually whenever the blood donation takes place, all right, or the transfusion takes place, if the blood is not sampled or checked properly, and if the samples are infected by HIV, then the donor, okay, who gives the blood is not affected. Basically, is an HIV patient. Whereas the acceptor, who even though is healthy, when he receives this infected blood, he becomes HIV positive. All right. Next one is sharing infected needles and surgical instruments, use of surgical instruments which are not sterilized properly. So at that point also you can get HIV. Okay. Next one, from infected mother to our child placenta. So this that means the HIV virus can cross the placental barrier and cause an infection, okay, to the babies also. Okay, next one. When we talk about in sexual practices with multiple people, but the problem is we wouldn't know which of the people would have the HIV. Okay, so at that point, a person who is having this multiple sexual partners would be vulnerable for or highly it's highly possible that this individual can contact AIDS. Correct? No. So multiple partners and mostly unknown partners are the cause for getting HIV. Next one, children who are born to the HIV infected mother. And there is one more way in which the community experiences the burst of HIV cases is through drug addiction. So in most cases, what happens when it comes to drug addiction, not all drugs, use of all drugs would lead to, HIV infection. There are certain drugs called as communal drugs like heroin. Okay, so heroin is shot into the veins. Like usually it is taken by injection of it into the veins. So the needle or the syringe that is used for the injection of the heroin, okay, is just shared among individuals in the community. So when it is communal drug addiction, so usually what happens is the people have high probability of getting this disease. All right. So this is about drug addicts. Okay, next one. So some of the people, like the people who are undergoing dialysis and the people who are going under dialysis or the people who requires blood transfusion on a regular basis, so they also have high probability of contacting HIV. Understood? So why we have HIV? There is one more part of it, one more aspect to it. The other aspect to study HIV is because if you observe in our community, more than the facts, we are driven towards misconception. So having this misconception against a particular disorder can put the person who is really suffering from this disease into a more worst part. There was a case in our state. Okay, so it was given in the news where a family, mother and father, are HIV positive, and the child is HIV negative. That means the child do not have HIV. So the villagers got to know that this family is HIV infected. So what they did was they banished these people out of the village. They were given no access to education, no access to food, or no access to any common or public places. Okay, and the child was also thrown out of the school. Now this misconception and uh, like what do you tell, this misconception and people who have very low knowledge or lack of knowledge will try to do certain things that might hurt the individuals who are already suffering from this disease into more worse case. Okay, it can turn into more worse case. So that is why we have to understand the ways in which we don't get HIV. Even though we can be around the HIV infected person or a positive person, there are some ways which you cannot get HIV. For example, if you kiss a HIV positive person, you will not get infected. If you hug the HIV positive person, you will not get infected. Similarly, sharing food, insect bites. Okay, insect bites. Like I told you in the beginning, like why mosquito bite do not spread HIV. Next one is sharing public toilet seats, bathing together, okay, sneezes and coughs, or even the sweat. So these are the possible modes in which a person cannot contact HIV. Okay, next. So this is why we have to understand how the HIV do not. Now, what is one thing that is more dangerous about HIV? See, once you make a contact with the individual, and if you get a disease in the very next few days, then at least there is some precautions. At least you are aware of what's happening, and you know that would really change the situation. Okay, but in HIV's case, HIV has a very long incubation time. What is very long incubation time? This disease takes a very long time for it to express once it is infected. For example, if a person gets infected today, okay, he makes contact today. It's not that tomorrow alone he gets the HIV symptoms. Tomorrow alone he shows the HIV symptoms. He might take few weeks, few months, or even several years. Okay, their incubation period can last from 5 to 10 years. So what is this incubation period? The period at which, for example, today I told, no, today is a person that is infected. Okay, so it has entered the body. Now from this day all the way till the day he starts showing the symptoms, and this duration between these can be months, weeks, or even years. This gap of days in which the virus or any pathogen resides in our body, okay, inactively, this period is called as incubation period. And the HIV virus has a very, very long incubation period. So that is what makes it even more deadly. And appearance of symptoms takes a very lot of time. And the people are usually astonished how they contacted HIV, because if they get after 10 years, who would remember? Correct? No. So that is another peculiar thing about HIV. Fine. So now we have to understand what is the life cycle of HIV. Okay, how does HIV cycle occurs in our body? Okay, so let's imagine through infected blood fluids, okay, or through any surgical instruments or syringes, etc. So once it enters the body, HIV particularly attaches or attacks, okay, a specific type of white blood cells called as macrophages. Okay, so once it attaches to the macrophages, it enters into its plasma membrane and even the cytoplasm. There what happens, the viral coat releases, okay, a special package into the cell, and this is called as a capsid. Okay, so let us assume this is the bigger virus. Inside it there is one capsid. Within the capsid there is single stranded RNA and all the enzymes. So this capsid is released into the, okay, cytoplasm of the macrophages. Once it is released, the capsid actually protects the viral genetics, viral genome, and then within the capsid there are essential enzymes and proteins that protects and also promotes the viral replication. So okay, so once the capsid is open, now the single stranded RNA is relieved, and the single stranded RNA is converted into a single stranded viral DNA through an enzyme called as reverse transcriptase. Okay, so we know that in the central dogma of life that the DNA is converted to RNA and RNA is converted to proteins, right? So here what happens, specifically the DNA is the RNA is converted to DNA. It happens in a reverse manner. So transcription happens ulta, okay, or opposite way. That is the enzyme that helps for this purpose is reverse transcriptase. All right. So now what happens? This viral DNA gets incorporated into the host DNA. Now, once it gets incorporated into the host DNA, now what happens? The viral DNA will start synthesizing more and more of viral proteins. Okay. So these viral proteins are produced in a very, very long chains. Understood? Now, these immature viral proteins are used for the construction of new viruses. Okay. So they're cleaved into smaller proteins, and these smaller proteins are used for the construction of new virus. So that's how the macrophages will become a HIV factory. Now, once the numbers are sufficiently increased, at one point, HIV enters into the specific type of lymphocyte cells called as T helper cells or helper T cells. So usually we tell the CD4 or CD8 cells. Okay, so these cells have a specific role to make your body, okay, to recognize the self and non-self cells, and also it gives an ability for our body to identify the pathogens and recruit a specific mechanism to defeat those pathogens. Okay, so without helper T cells, we cannot produce antibodies. So once this HIV virus attaches to the helper T cells and starts its replication and production of progeny viruses, okay, through the same process of reverse transcription and viral DNA incorporation. Okay, now what happens? The reduction of T cells happens in a very tremendous way. So if the number of T cells keeps decreasing, then our immune system will get weaker and weaker, and eventually it becomes defenseless, leading to a syndrome like condition. So this is about HIV and how it manifests in our body and how it divides. All right, so once we have this sufficient reduction in the number of T cells or helper T cells, it leads to certain symptoms. Okay, it shows up. The symptoms start showing up. So what type of symptoms can we identify? So during this period where the person starts showing the symptoms, they show regular fever. Okay, occurrence of fever is very regular. Diarrhea. Okay, weight loss, deficiency of T cells. Okay, or they usually get infected by Mycobacterium. So Mycobacterium tuberculosis is a pathogen that causes tuberculosis. So tuberculosis is a very deadly disease. Okay, or it might be pleural or lung tuberculosis or intestinal tuberculosis. All these different types of tuberculosis when a person starts showing, and they might also be infected by viruses, fungi, parasites, and even toxoplasma. Okay, parasites like toxoplasma. So usually these persons show more symptoms rather than one particular symptom. They show multi-symptomatic conditions. So how do we detect that a person who is having this particular HIV? So we use a kit called as antigen testing kit, and this kit name is called as ELISA. ELISA stands for enzyme linked immunosorbitase. So what do we do here? We try to target a specific polysaccharide, okay, or a protein on the HIV protein coat. Okay, so these our antibodies will try to identify them and attach to their viral surfaces. That will lead to the precipitation. So when it precipitates, that will show the presence of HIV virus with a change in color. So the person who precipitates and changes color, it is called as HIV positive. And the people who do not show any change in color, they are called as HIV negative. Followed? So this is about diagnosis, and the diagnostic kit is called as ELISA, and it is an immunological based assay. Remember this. Okay. So what about the treatment? So there is no specific treatment for HIV. So what do we do? Just remember, you can apply your own knowledge here. So if a virus comes into our host, it has to attach to the cell surfaces. Okay. So we can build something that hinders the attachment to the cell surface. So that is one way. Second way is by stopping the reverse transcriptase, and the third way is by stopping the incorporation of the genes. Okay, so these properties are called as antiviral properties, and the drugs who does that is called as antiviral drugs. So they are not 100% effective, but they can prolong the life of a patient for a specific period of time, thereby relieving them out of this entire HIV situation. Okay, fine. So the most important part of HIV and the control of this disease is by educating. So how do we prevent this? So some of the ways you can prevent this is by educating people about what is AIDS and how does it spread and how do we prevent them. Next one is avoiding consumption of drugs, especially communal drugs, which involves the use of syringe. Okay, so we can control drug abuse. Next one is use of disposable needles and syringes, especially in the hospitals, including the public or private sector hospitals. Next one, advocating about safe sex, like use of contraceptives, like physical barriers, like condoms. Okay, avoiding multiple sexual partners and unknown partners. Okay, next one, free distribution of condoms. Usually it used to happen in recent times, where government used to distribute free condoms and public places like bus stops, railway stations, hospitals, etc. Next one is regular checkup for HIV. As I said, HIV has a very long incubation period, and it is highly necessary for an individual to, go and get themselves checked periodically. Okay, so this is about a regular checkup of HIV. Next one is making blood banks safer from HIV. So this is quite tedious task, but our blood banks can go under extensive screening processes. Okay, before they infuse the blood into the patient. All right. So next topic is cancer. So we might have heard about cancer a lot of times, or even seen the situations where some of our people who are very close to us or who are around us might be suffering from cancer. Okay. So what is cancer? Uh, uh, how bad is cancer? So cancer, in simple terms, is abnormal or uncontrolled multiplication of cells. Okay. So you just have to look at yourself. Uh, you don't see an excessively larger head on your shoulders, or you don't see having 20 fingers in your hands, or you don't see 5 to 6 eyes, or you don't see that each and every human having like completely weird morphological characteristics. It doesn't happen, right? We almost look similar or exactly same. So this property of us showing this human-like characteristics happens because of our genetics. Okay, our genes knows how to construct our body and how to organize our cells, or how many cells have to fit in our body. So at one point, what happens is the failure of the genetics, or because of some reasons, our cell becomes very abnormal and uncontrolled multiplications. Okay, uncontrolled multiplication means they can divide without having any breaks, no break. Okay, continuous divide, 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 divide. So this uncontrolled multiplication, what does it result? It result in a huge mass, correct? No. So if my fingers, or if the cells near my throat or brain or anywhere, if these cells start dividing and they're not answering to any of the body's signals, so what would happen? It would cause the formation of a mass. So this mass of cells, okay, which is a resultant of uncontrolled division, this we call it as tumor, correct? No. So this will call it as tumor. So what is the difference between what stops the normal cell from dividing and what does not let these cancer cells to stopping dividing? So the reason is the normal cells, as they divide, they really communicate with their neighbors. Okay. So as they keep on dividing, there will be a point where our cells will come in contact with the neighboring cells, and then it prevents them from dividing as they come in contact with them. So this prevention of the cells from dividing just because of their contact with the neighboring cells, this property is called as contact inhibition. So that is what helps us to know how exact amount of cells have to divide into a particular tissue, in a particular organ, or a particular part of our body. Okay. So usually cancer cells do not show any of this property of contact inhibition. Fine. So let us see what are the two different types of cancer. The types of cancers are, one is benign cancer, other one is malignant cancer. So let's see what is benign cancer and malignant cancer. Benign cancer though are those type of cancers where it is localized cancer. That means it is only restricted to a specific part of the body. Okay, so it is confined to an origin. Where it starts, it's going to remain there. Understood? So these are called as benign cancers. So the property of benign cancers is they do not spread. Okay. If it is pancreatic cancer, they're going to remain in pancreas only. If it is colon cancer, they're going to remain in colon. Okay. If it is liver cancer, they're going to remain in liver. Understood? So these properties of being to a confined place and not having the ability to spread, okay, and it causes a little damage. Not compared to the malignancy, it only causes a little bit damage. So this is called as benign cancer. All right. So one second. Hold on, students. Yeah. The next type or malignant type severe compared. So what happens? There is a proliferating because of tumor causes. Now, if some of the cells of this proliferating mass, okay, detaches, okay, if they detach from the local area, so and then they spread through the blood and attach to the new location and start a cancer there. So this property of the cells detaching from the mass of cancer cells, moving to the new place, and then restarting the cancer properties, or restarting the cancer in a new organ or location. So this is called as metastasis. Okay. And the cells which detach from the mass of this cancer is called as neoplastic cells, or it is also called as tumor cells. Understood? So they grow very rapidly and they are highly invasive. Okay. So they damage the surroundings. Usually what happens, the cells or the mass, they integrate or they connect themselves with the local neural tissue as well as the blood vessels, and then they are highly competent for the nutrients. So when they're highly competent for nutrients, they'll make the normal cells very deficient. Okay. So that is why the person will lose a lot of weight when they're suffering from cancer. All right. So who causes cancer? Anything that are called as carcinogens. Okay, 1 second. Okay, anything that causes cancer is called as carcinogens. All right. So carcinogens can be of several types. They can be physical, they can be chemical, as well as they can be biological. All right. So let us see what are the physical agents. For example, if a person is exposed to ionizing radiations like X-rays, gamma rays, okay, and even the non-ionizing radiations like UV rays, okay, they can get cancer. Okay. So when it comes to chemical agents, something like, you see the advertisements on the TV, or when you go to the movies, right? Don't smoke tobacco. Okay, dhumrapan, arogya ke hanikar hai. Okay, or smoking cigarette or consumption of alcohol causes cancer. Have you seen these advertisements? Yes. So exactly. So this tobacco smoke contains nicotine, okay, which can cause cancer. And next one is vinyl chloride, caffeine, okay, mustard gas. These are all some chemical agents which have the capacity to make our cells behave in a cancerous fashion. Understood? Next one is biological agents, something like oncogenic viruses, okay, cellular oncogenes or proto-oncogenes. Or proto-oncogenes are the genes which can cause cancer. Okay, so ornithine normal cells is activated whenever, and that they become oncogenes. Okay, so these cells become oncogenic. Okay, so these agents which can cause these cancer situations are called as carcinogens. So let us see, how do we detect the cancer? So cancer can be detected by several techniques. One is physical techniques like biopsy, imaging, use of antibiotics, and even molecular biology techniques. So what is a biopsy? So usually in certain regions like brain or in colon, okay. So what they do is they send a laparoscopic tube, okay. So as they send the tube with the camera, they go to this location where the tumor has occurred, okay, through a very precise surgical instruments. They isolate some of these cancer cells from their location, and then what they do is they conduct histopathological assays. Histopathological assays is they grow these cells in the laboratories and understand how the cells behave. Okay, either they are malignancy or they are benign cancer or localized cancers. How fast is the growth? Everything can be studied in the labs. So this histopathological assays would give us confirmation about the behavior of the cancer and what type of cancer, how vigorous is cancer, etc. In many of the cases like leukemia, that is blood cancer, what do you call it as, where there is excessive number of white blood cells. Okay. So next one is blood and bone marrow test can increase can be seen to check the cancer. So cases like breast cancer, lung cancer, and colon cancer, and you can see liver cancer. For all these things, biopsy is conducted. So this is one of the very promising ways to examine what type of cancer it is. Understood? Okay. Next one. Hold on. Yeah. Yeah. Next one is radiograph techniques or imaging techniques. So you must see one of the basic ways to recognize the mass of cells is by taking the X-rays. So how is it even possible? If you see the normal, if you're not a medical technician, you would understand that there is some difference in the imaging. of these two individuals, and this is not normal. So similarly, as you become more proliferant in this knowledge, easily we can understand that there is something that does not belong to a particular region. So that is what imaging does. So basic imaging is X-rays. Advanced imaging is, okay, CT scanning. CT scanning is computerized tomography. Here what happens is several sections, okay, of this rays are passed into your particular organ like brain or any liver or any lungs. Okay, so these sections, what it will do is it will make your body, make your organ into very, very, very thin sections, like 1,25,000 sections are taken, then they're all compiled together. Okay, and it produces a 3D image. So it's very fascinating, to understand this. So that is called a CT scanning. Next one is MRI. MRI is magnetic resonance imaging. So when the magnetic field and non-ionizing radiation are produced, here what happens is, through MRI we can see how much the tissues are integrated with nerves and the blood vessels, how many blood vessels are integrated, how deep is this cancer that has spread through the internal organs. Okay, all these factors can be understood very clearly using MRI. Okay, next one is this is even more quite advanced studies where we use antibodies. Okay, some of the antibodies are designed to specifically go and attach to the cell surfaces of cancer. Okay, cancer cells. So this will create some kind of a precipitate. Okay, so showing that, okay, a person has a cancer. And molecular biology techniques, like you can use your PCR techniques, okay, or even you can use several other techniques in molecular biology where you can detect the cancerous genes. Okay, so some of the ways in which you can prevent cancer is by stop smoking tobacco. Okay, and by preventing your use of alcohol, excessively going in the sunlight, okay, without any protection. Okay, so all these things are very, very necessary that you have to follow to prevent cancer. Okay, so how do we treat cancer? Some of the ways, there are, it's not just one way that we can choose. We cannot tell that, okay, I will choose one medical technique to cure cancer. So there is several integrated techniques are applied to use, for the treatment of cancer. So some of the ways in which we use, some of the techniques that we use to treat cancer is, first one is radiotherapy. So under radiotherapy, what will happen? The tumor cells are irradiated. Irradiated means they're exposed for the lethal radiations, and usually the surrounding tissues are protected from these radiations. Okay. Next one is chemotherapy. In chemotherapy, some of the chemotoxic agents are used, and many of these drugs are very, very harmful, and they can cause several side effects to our body, such as hair loss, anemia, lack of appetite. So that's why the people undergoing chemotherapy, they have loss of hair in the head, lots of hair in the eyebrows and eyelashes. So they lose a lot of hair like this. Okay. Next one is immunotherapy. So immunotherapy is a specific, antigen binding antibodies are given where the target these, they target these cancer cells and destroy them. And another way is like something called as biological response modifiers, such as interferons are used, where it activates the immune system of the cells to destroy the tumor. And the final one is surgery. Surgery is one of the common ways to treat cancer. Okay, where the mass of the cells are relieved, removed by incision. Okay, so this is called surgery. As I said, most cancers are not cured by one methodology. Several techniques are combined for the treatment of cancer. Okay. All right, students. Our next topic is drug and alcohol abuse. So why do we have to study drug and alcohol abuse? So basically, in our society today, everybody, most of the people are quite affected by these distractions that happen either now or in the coming future. So many of the situations where an individual may not be interested, but they might be exposed knowingly or unknowingly to certain drugs. Okay, so that is why, even though you want to do it or you don't want to do it, or sometimes you might be accidentally coming across it, knowledge about it or knowing about it would really save many lives, and it might help you to prevent any of such addictions or any of those temptations in the near future. So in our community, one of the most commonly used drugs come from three most important categories. One are opioids, cannabinoids, and coca alkaloids. Okay, so let us see what are opioids. Usually, opioids are a certain type class of drugs which can disrupt the central nervous system as well as gastrointestinal tract. Okay, some of the examples of opioids are morphine, heroin, and brown sugar. Okay, so each of them are, they have a specific way in which they are taken. So let us see about morphine. Okay, morphine is usually extracted from the latex of a poppy plant. You can see that there is poppy plant, and poppy has a poppy bud. So they cut this poppy, buds, and what will happen? It will start lactating, a latex out of this poppy. So this latex is collected, and then it is basically converted to morphine. That poppy plant is also called as pepper somniferum. So it is used in medical sciences. Basically, when a person is going post surgery or when they're in excessive pain. So it is a powerful sedative and a painkiller, and it is used more in medical surgeries. Okay, next one. Next one is heroin, and it is one of the most dangerous drug, and it is very very addictive, and it is highly impossible, very very tough and impossible to come out of heroin addiction, and it is one of the most feared drugs and highly addictive drugs, I can tell. So heroin is also called as smack, or it is also called as diacetyl morphine. So the color of heroin is white, and it is odorless and bitter, and it is a crystalline compound. So it is obtained by acetylation of morphine. Okay, additional of one more acetyl group would give you diacetyl morphine. Okay, so how do people consume morphine? Morphine is either consumed by snorting. Snorting means taken through nose, and or by injection. Understood? So how does a morphine affect our system? So basically, it is a depressant. It completely slows down a person, and it will put them down into this euphoria situation where they are knocked out of senses and they don't want to do anything, and they just remain numb and lost in a place for a very long time. And it slows down our body function tremendously slow. Okay, so usually when people undergo surgery, okay, after they come out of anesthesia, and if they are in excessive pain, the morphine is given. So when the morphine is taken, that's why the patients sleep a lot, and they are not responding to you properly. Their cognitive functions are very very poor, and they seem very slow. That is because the patients are given morphine. Understood? So the next class of drugs is cannabinoids. Okay, it's one of the most used drugs, very cheaply available, and it is the most abused substances in our country. So fine. What about cannabinoids? Cannabinoids, like these are cannabis. They belong to a class of plants called as cannabis indica or cannabis sativa. So when the cannabis is smoked or eaten or consumed in any way, so they have a specific chemical called as tetrahydrocannabinoids, which go and bind to the receptors, tetrahydrocannabinoids, that is THC receptors in the brain. So after they bind to the brain, usually the people undergo this sense of euphoria, that is goodness, slowness of time, okay, increased appetite, okay. So some of these are the symptoms of using cannabis, okay. So how do we obtain cannabis? What parts of the cannabis are used? So you can see in this picture, the buds or the inflorescence of this cannabis is used for smoking, okay, or it is used for making some of the cannabis byproducts like hashish or charas, okay. Next one, usually some of the flower tops, leaves, raisins can also be used as marijuana or hashish or charas or ganja. So some of the people who consume marijuana also report that their heart rate increases. Okay, so it basically affects the cardiovascular system. Okay, and as I said, one of the most abused drug around us is cannabis. Okay, and it is used by sports person. All right. So have you heard about cocaine, or some of the most commonly used term in when it comes to drugs, cocaine, correct, right? So you might have seen in the series, or we would have heard somewhere. So cocaine is a drug that is quite addictive. Okay. See, some of the drugs, even though when you read an article, it might show that they do not have side effects. But when you see the other portion of these drugs, they show that they're highly addictive. So highly addictive part of these drugs can affect you very severely. So we'll talk about it just by finishing this cocaine part. So cocaine is extracted from a plant called as Erythroxylum coca. Okay. So cocaine, what it does is it goes and interferes in the neurotransmitter, that is dopamine. And as this transport of dopamine is interfered, now the person feels highly energized, euphoric, and more confident, okay, and bursted with a lot of energy. And the ways the people use cocaine to consumption is either they snort, okay, the most common way is by snorting. And some of the cocaine is also converted to a black color substance called as crack, which is more popular in US drug culture. It's worse, like 100 times worse than cocaine, consumption of cocaine. So it's as it stimulates the CNS, and euphoria and increased energy is felt by the individuals who use cocaine. That is why whenever the people who use cocaine, they don't know how much doses they're consuming. So excessive dose of this cocaine can lead to hallucinations or even cardiac arrest in severe cases. Okay. So, and some of the hallucinogenic plants can be Atrofa belladonna, datura, and it can be some of the drugs like barbiturates, okay, amphetamines, okay, benzodiazepines, etc., can also be used as drugs. Okay. So these drugs like barbiturates, amphetamines, and benzodiazepines, they're used to treat mental illness, depression, and insomnia, etc. As a result, these people start using for their own enjoyment and recreation. Okay. So whenever these drugs are abused, it can lead to physical, physiological, or psychological impairments. Understood? Fine. And the last part is tobacco, right? Smoking tobacco. We have been advertised a lot. We have been educated a lot. Okay. So when it comes to tobacco, it's been used as a cultural drug for over 400 years until the time of cultivation. So we know that tobacco is either smoked, chewed, or snuffed. Okay. It contains something like nicotine, substance like nicotine, and alkaloid, which stimulates our adrenal glands. So the release of adrenaline or noradrenaline increases our blood pressure and heart rate. Okay. So that is why people smoke a lot of tobacco. So usually smoking is really bad. Why? Because it causes lung cancer, urinary bladder cancer, throat, emphysema, bronchitis, coronary disease, coronary heart disease, and even gastric ulcers. Okay. So tobacco chewing usually causes oral cancer, and smoking increases sometimes carbon monoxide content in the blood, which causes carbon monoxide poisoning, and it reduces oxyhemoglobin that causes oxygen deficiency. So usually we call this condition as emphysema also. Okay. So in the next class, as the final part, we will study what is adolescence, what is drug and alcohol abuse, and prevention of this. All right, students. So if you have any doubts, you can post the doubts in the chat box. I'll be here, or you can carry your doubts in the next class. We can solve these doubts. Okay. Thank you very much. Have a nice day.